We're going after heart disease. That's 100% of what we do, leading cause of death in the world. After a single infusion of TN-201, we were able to show that 6 to 12 months after dosing, all the patients, their hearts started to normalize. When it works because it's a lock and key.
Our coverage of Meeting on the Med in Rome is kindly sponsored by Solaris. I am Federico Citteric, reporting for Onyx live here in Rome at the Meeting on the Med. Today I took some time to talk with Faraz Ali, board of ARM and CEO of Tenaya Therapeutics.
I'm the CEO of Tenaya Therapeutics. We're based in California. We're sitting here in beautiful Rome, but the company was founded and is currently located in the San Francisco Bay Area in California, established in 2016. So we're about 10 years into our existence, going after a pretty bold and exciting vision. We're going after heart disease. That's 100% of what we do, leading cause of death in the world, more than all oncology combined. And we're going after it with a bit of a unique approach. We are going after both rare and prevalent forms of heart disease. And one thing that really sets us apart is that we're modality agnostic. So in our current pipeline, we have clinical stage gene therapy assets, clinical stage small molecule assets. We have cardiac regeneration, gene editing. We recently announced a collaboration with Alnylam where we're working on other modalities as well. So a lot of different modalities. We follow the science, all on the heart.
What do you think makes the area of heart disease compelling in the context of gene therapy?
Well, let me take a step back and say what makes cardiovascular diseases compelling, period, and then we'll talk about gene therapy. When I joined the company as CEO about now approaching 8 years ago, what got me excited was the belief, the prediction that cardiovascular disease was going to go the same direction as oncology. And what do I mean by that? Oncology has made tremendous progress and strides in the field of precision medicines. Right? So we now stratify the different cancers based on sort of different markers, usually genetic markers, and then you have medications that are really targeted to those particular gene, you could be triple negative of that, HER2 positive for that. We don't just talk about breast cancer, we don't just talk about colorectal cancer, we talk about specific subgroups. And I felt like that's the direction that cardiovascular disease was going to go as well, right? It's a leading cause of death in the world, but we are now able to stratify that based on phenotype and genotype in a way that was not possible before. And that's only accelerating. So I just was excited about this opportunity to take a page out of the playbook of oncology and apply it to the leading cause of death in the world. So that was the general thesis. And that's partly why we're modality agnostic. We figured, well, depending on what the nature of the disease is and depending on what that stratification looks like, you may need a different medication. So for something, it might be gene therapy, but for something else, it might be a small molecule. It really depends. So that's sort of the big vision. Now, gene therapy within that, we now have, as we do in oncology and many other therapeutic areas, a much better understanding of some of the genetic drivers of disease. And I would put that into two categories. One is really canonical, very severe, genetically driven diseases, and that, these are often people who are presenting with severe heart disease in their young adulthood, even in their childhood, even infants. And that's because they've inherited one or two genetic mutations that is clearly driving the underlying problem. And those are really, those are very amenable to gene therapy or gene editing, right? So if you have a defective gene, you may either have to add a healthy copy of that gene, or it may make sense to edit that gene. Now, which one, it depends. But so that's where gene therapy is exciting, because when you know the true genetic driver of the disease, then and where you know that a defect of the gene is what's causing the problem, then you need to fix the defect or add a healthy copy, and that is the field of gene therapy and gene editing. And so that's something that we're doing today. There's another side of genetics in heart disease which is also interesting, which is larger indications where you have a SNP or you have a marker that seems to predict a faster progression, but it's not necessarily going to be addressed by a gene therapy. It's too large of an indication, or the risk-benefit ratio of a gene therapy or gene edit isn't quite there. but it's still helpful for stratification. And for those, you might do more traditional modalities like small molecules or antibodies or siRNA or something like that versus a gene therapy or gene edit. So we do both. We're involved in all of that. Almost everything we do is firmly grounded in some understanding of a genetic driver of that particular condition.
And what are the challenges, on the other hand, in the area?
Well, first, the heart is a fascinating and one of the most complex organs, right? It is uniquely an organ where there is both electrical signals propagating across it and mechanical movement, right? If you think of almost any other organ in the heart, it might have mechanical movement, like say the lung, or you might have electrical firing, like the brain, but the heart, you have both. And it's actually actively pumping blood away from it to the rest of the body. So one of the key things that is challenging in the context of drug development for heart disease is trying to deliver to the heart and try to do it in a way that doesn't mess with, and in fact hopefully improves, this very complicated interaction of electrical and biomechanical movement that is happening across the myocardium. So that's what's exciting and interesting and difficult about it. In the context of the heart, delivery and genetic medicines for the heart, the key thing is delivery. Right? How do you get something into the heart that won't get pushed away? And the primary modality that has emerged is AAV. AAV, or adeno-associated viruses, are the workhorses of the genetic medicine space for the heart. You infuse it and circulates around the body, but then a good amount of it will end up going into the heart and penetrate the right cells of the heart, and over there it'll express itself, either the healthy copy of the gene or the gene editing machinery. So I think the complication is delivery, and the solution to that is AAV. We happen to be, for our 2 clinical stage programs, we're using AAV9, which is the most widely used capsid in the world. More than 5,000 patients have been dosed worldwide with different kinds of AAVs, commercially approved AAVs, for severe genetic diseases. And it turns out that those capsids also work well at penetrating the heart, both in terms of entering the cells of the heart as well as expressing the cargo, whether it's a gene or a gene editing machinery.
Faraz, do you think that working in the field of heart disease, which is of course an enormous field, changes the way you approach development and scale?
Not only are we a therapeutic company, but we have invested significant resources in the manufacturing side of things. How do you make capsid? How do you make manufacturing possible so that you can scale up and bring down the cost of goods and produce enough material potentially for large indications? So we've done innovations not only on the therapeutic side, but we've actually done innovations and have patents on the manufacturing side in order to try to solve some of the scalability issues that are inherent with taking something like AAV to larger indications. For some of the other programs that we're working on that are not AAV gene therapy or gene editing, for example, a small molecule, no problems with scale.
Yet the field of cardiovascular gene therapy is perhaps less crowded. So what are you looking for in conferences like this, such as MOMED?
So wait, I'll take a step back. Why am I here? I'm here because I'm also on the board of the Alliance of Regenerative Medicine, which is an outstanding organization that is the organizer of a conference like this, the meeting on the Med, and they also do a meeting on the MESA, and many, many other things to advance the field. So I'm here as a capacity, partly as a board member, where we get together, several times a year, as any board does, to think about the health of the field and what are the things that we need to do. Do we need to do more investment here? Do we need to do more advocacy there? Do we need to engage with the FDA? Do we need to convene people who may be competing, but they need to work together to solve some problems? That's partly why I'm here, is because this meeting was organized by them. Now, I'm also here as a therapeutic developer. Tenaya Therapeutics has products, right? We just talked about some of them. And so we're interested in engaging with others, potential partners, learning from others who have had some success or had some setbacks, talking to potential collaborators in either research or development or manufacturing. There's a lot of suppliers over here of different services. So I'm here also for that.
Faraz, could you please tell us a bit about your lead gene therapy program?
Yeah, great. So we have 2 clinical stage gene therapy programs. One is called TN201, the other is called TN401. TN201 is going after the leading genetic cause of hypertrophic cardiomyopathy. That's a terrible condition where, as a result of the mutation in a particular gene, the walls of the heart start thickening and the overall heart starts enlarging. There's fibrosis, there's arrhythmia, there's over time these hearts start to fail, and many patients eventually progress to the need for something like transplantation. The more severe version of this disease affects kids as young as the first few days and months of life. You know, they're kids who are born with 2 mutations, and some of them die within the first year of life, universally. So there's no medication on earth that can really help some of these really severe patients. And what we are simply doing is adding a healthy copy of the gene. And what we've demonstrated in humans is that you can take patients who, despite every standard of care, medication or therapy or surgery, their hearts are still enlarged, their walls are still thickened, there's still dysfunction, and it's actually getting worse. And what we were able to demonstrate, and we presented at the American Heart Association last year, is after a single infusion. These are, remember, gene therapies are often quote unquote curative. Prefer not to use the term curative, but single dose. After a single infusion of TN201, we were able to show that 6 to 12 months after dosing, all the patients, their hearts started to normalize. Certain blood-based biomarkers were normalizing, but more importantly, we started to see that the walls that were thickened start to thin, and the overall size started to shrink, and that was something that had never been seen in a patient population like this before, that extensive degree of remodeling after a single dose. And of course, the patients were also feeling better. So they were New York Heart Class 2 and 3, and now they're New York Heart Class 1. That's just basically a way of capturing, are they burdened by their heart disease? And we were eliminating that burden. So it was a remarkable early stage still. We were only talking about a handful of patients, but it's given us that reason to believe that, wow, this stuff is actually working. And we're the first company in the world to try to tackle this very severe genetic cardiomyopathy with a gene therapy. So that's TN201. TN41, very different. It also is a genetic cardiomyopathy, but over here, the hallmark of the disease is not thickening of the ventricles and the enlargement of the heart, but it's profound arrhythmia. These patients have terrible electrical instability, and unfortunately for about a quarter of the population, the first presentation is sudden cardiac arrest leading to sudden cardiac death. So by the time you find out they have the condition, for about a quarter of the patients, it's too late. They're already dead because somebody didn't get to them in time when they had a cardiac arrest event. Again, we understand the genetics of this really well. It's a mutation in a particular gene called PKP2, and that results in all the downstream electrical instability. And what we're doing with TN41 is adding a healthy copy of that gene using AV9, as we described earlier. And after that single infusion, again, we also presented last year, we were able to show that in the first few patients that we dosed, they went from profound electrical instability to dramatic reductions, approaching something that you might call in the normal range. And so that, again, has never been seen before. These patients are on standard of care medications. They've had many surgical interventions like ablations, similar to the other program. And despite all of that, they were getting worse. And so a single dose, and 6 to 12 months later, we start seeing dramatic results like that. So we're so far batting 100% on both of our first 2 gene therapy programs. The first couple of patients we've dosed are doing they're seeing a response to gene therapy of a kind that has not yet been achieved with any standard of care medication or surgical intervention. Tremendous unmet need. And so it's tremendously exciting as a company, and for us to finally see years of work finally begin to pay off. And of course, we're still early in development, more to come. This year, we're going to be presenting more clinical data on both programs. We're very excited to do that, and we'll be engaging with the regulators about a path to pivotal studies and approval. That's one beautiful thing about gene therapies. Things can go, they're harder, but things can go fast. When it works because it's a lock and key. You've got a genetic defect, you're adding a healthy copy of the gene, and that has historically, so far in development, led to pretty dramatic outcomes, which has convinced the regulators we got to accelerate the path to approval and access for patients. And that's a ride we hope to go on with these two programs. And this year will tell us a lot about how close we get to that mark.
Well, Faraz, thank you very much for joining us. It was a pleasure. Thank you again.
Take care.